Could you unmute yourself, Aykut? You are muted.
Complain. Okay. Can you hear me now?
Yes.
Yes, good. Okay, great. So why does it matter to study anomalous diffusion? Can you give me an example of a Brownian motion, the standard Brownian motion? Anything that is in the real life that is operating with standard Brownian motion? Not really, that is anomalous diffusion. Any other examples that is having a standard Brownian motion? Yes, dust particles in air or synthetic particles in a liquid environment, very artificial, experimentally created environment. But if you can think about anything that is in real life, cells, animals, birds, fish, all of these things don't really operate with the standard Brownian motion algorithm. Most of them have some kind of randomness in their motion, but they're still not really operating with the standard Brownian motion. They don't say, okay, I flip a coin now. A bird doesn't do that. I go right, I have to follow this kind of universality class because the mean of my motion should be 0 and the standard deviation should be. They don't operate like that, and they're complicated, such as these birds. Anyone from here near fowl shopping? Anyone? No? This is a place near fowl shopping. I forgot the name of the lake, but these crane birds are passing by there every I don't remember, April it can be. And these are a standard example of anomalous diffusion. They make a random motion, but they also make some kind of flights for their route, right? And then whenever they arrive at some place, they kind of do to make random motions for a while. When they camp here for a few weeks, what they do is going around, flying one way or another, right? They're looking for food. It's the same with fish, for example, in the ocean. They have some routine, they follow some trajectory, they have a general idea of what they're going to do, but in most of the time, their motion is still random. They're looking for food, right? And what they do is they go in one direction for a while, they go in another direction for a while, they're looking for food. Their motion, while being still random, it's actually in some time scales, it's not totally random. They also immigrate, for example. There are birds that immigrate to south in winter and come back in summer. And that's what we are going to discuss in this lecture, okay? And I want to make, to understand the anomalous diffusion, there is one important concept that I want to introduce, which is called ergodicity, okay? Which means that the ensemble average of a system is identical to the time average of the system. So it's time symmetric what I'm doing. This might be very complicated to understand, but I can simplify the mean square displacement calculation in two different ways. What is the mean square displacement? The average square displacement I have, right? I can calculate it with two ways. One is what I'm calling ensemble mean square displacement. Okay, I have, for example, 100 birds, okay, that start at one position, and I check after every time step where each individual has gone, and then I check the square displacement of each individual and make an average. This is called an ensemble average. Okay, what you do here is that this is xi, and you take the xi here is the individual bird, let's say, or a particle that we are analyzing, and you average over all the different particles, and you start with a certain time, and you kind of, the tau doesn't even matter here, because you kind of take this 0. This is the beginning, and you can even assume that the beginning, you can scale or translate your system such that your initial point is assumed to be 0, and then you are just taking the average squared of all the particles for different time steps. Okay, so you are having many particles. Think about those individual trajectories I was showing you in comparison with different time step simulations in the previous lecture. I have 1,000 trajectory. I take the average of x squared for each one. Okay, is this clear? How is this numerically treated? This one is maybe worth demonstrating. Okay. So I have, let's say, an ensemble which is capital X. I have many particles that have trajectories that are represented. Each one is represented by one array. Okay. What I do is that I take for time t, I take the difference between these two elements and take the square. Do the same for this array, do the same for this array, do the same for this array, and take the average over the ensemble, over the many different realizations of the same equation or system. Okay. If we are an experimental system, we have many particles and we calculate these square displacements for each one for different times, and then we take the average, which is called the ensemble average. Okay, is this clear? This is the concept. And there is another way we can calculate the mean square displacement, which is the time average. Instead of having these ones, we have a lot of single trajectories that belong to different particles. Now for the time mean square average is that we have we have only one particle and it has a very long trajectory. Okay. What I do is that I want to calculate what is the average square displacement in t time steps. I treat the problem like this. I check this value. Okay. The square difference between this one, and I check this one, and I check this one, and I check this one. I go forward in the trajectory. What I do essentially is the following. So I take part of the trajectory, right, and assume that you can do it as long as you want. Of course, this has a limitation. I will show you just in a second. And you shift the whole thing by t amount, and you kind of take this difference between these two from the same trajectory and take the square of it, because this will be all the differences with t time steps of the particle through its trajectory, right? Is this clear? If I subtract these two parts of the same array, I will get differences, the displacements the particle has made in time t. Of course, I can change the t and repeat this calculation, but if I can make this subtraction and take the square of it, as indicated here, and now take the time average of it, because now I have a square displacement for all different times if I take this difference, and then I make an average over this array, which is the time average. So this is two different calculations. Of course, here n is the total amount of elements I have in my array at the end, so I'm averaging. Okay, so I'm just summing first and divided by the number of elements I made the sum. And these are two different methods of calculating or quantifying the mean square displacement of a trajectory. Is this clear? So I can either calculate the average over the ensemble, different realizations, or I can calculate the average over time if I have a very long trajectory. And for ergodic systems that have symmetry between ensemble and long time scales, which means that it kind of explores the entire phase space that it can discover, kind of, if you have a long trajectory. The standard example, very easy example of this, is a standard Brownian motion or diffusion, if you name it. So yes.
Did you say there were n time steps or n individual?
It depends which one are you calculating, right? So here, and if you are taking the E ensemble average, you have many trajectories, right? You take the difference between from the first value for all of them and you add them up. Here, your n is this, right? Now, if you are taking the time average, then you are averaging over this array, right? And then your n is this. So different n's there. It's just the same. How you are making a sum, right? That is the how many elements you are summing. That's your n, and they are different for both cases. And here you can see that we have a Brownian particle in overdamped time scale. So here the system is not inertial because it's quite random because the time scale is long. You can see here it's beyond milliseconds. It's actually going above a few seconds, the trajectory. And here, the circles represent the time average. What is a time average? Time average, I simulate a very long trajectory and I make this kind of an average with the mean square displacement of the particle. Okay? And what is the ensemble average? The dots. I simulate the same particle many times from the beginning and take the mean square displacement for each one and make the average. So average over many realizations or average over time from a single realization. Okay, that's the difference. And you see that for the Brownian motion, they're identical. Even for the very low time scales where you see that the particle here was moving micrometers, here it's only moving nanometers and time scales are sub-microseconds. What's happening in these time scales? Particles mass matter. You see that the motion is more consistent because the velocity cannot change immediately. There is inertia taking place here. And even in these time scales, the mean square displacement is the same if it's calculated from whether the ensemble average or time average. Okay? Is this clear? And you can also take the optically trapped particle. It also has a mean square displacement. Of course, what happens if we have a freely diffusing particle to mean square displacement? Like a random walk, it's increasing linearly with time, right? What happens if I trap this particle? The square displacement cannot increase more than a certain value because it's trapped. So it cannot go anywhere outside of this region, right? So that is why it's increasing in the beginning, but it is saturating to a value of the variance of the particle trajectory in this trap after a while. But still, whether I calculate this mean square displacement over the ensemble, different realizations, or over time for a single realization, is giving me the same results. Okay? Yes.
What is the dashed line? Black dashed line.
Black dashed line is the freely diffusing particle. The result. No, the black dashed line is not that. The black dashed line is slope 1. This is just for comparison. So the position of this is not important. Its slope is just here to represent. This is the mean square displacement trend, the scaling of standard diffusion to compare. So you see that the particle in the inertial time scales, its mean square displacement is increasing higher than the slope. This is slope 1, which means that the black dashed line is representing mean square displacement that scale linearly with time. Of course, if you have a particle that moves with constant velocity, how does it scale with time? Its mean square displacement. If I have a constant velocity, I am going with v, right? My displacement after time t is vt. My square displacement is v squared t square. So it's scaling with t square instead of t. So it's higher than, of course, t, which is what you observe here, because at very short time scales, this particle's velocity is kind of constant because of its inertia. So its mean square displacement is scaling higher than t because its motion is more ballistic than random, right? And as you increase your time scale, it goes to a regime or approaches. Here it's the continuation of this, if you will. You see that here is 10 to minus 5, 10 to 0. You can actually make the simulation, this one, a lot longer, and you will observe actually that you are going to have like very like ballistic like motion and these time scales, and it will converge back to linear parallel to this, just as in this case. This is what we were showing in the inertial to non-inertial Brownian particle case. Okay, is this clear? And of course, this doesn't, this kind of standard Brownian motion doesn't really model all the different behaviors in the Earth. So that's why in the nature. So we need different models, and different models indicate different. So the Brownian motion is the model is sampled linearly in time, right? We calculate the x in time is equal to 0, and then time is equal to 1, time is equal to 2, time is equal to 3. So my time scale is always linear. But most of the anomalous diffusion models have time scalings that are non-linear. The reason is that they allow them to get a little bit more freedom of complexity to model different kinds of behavior of motion. And this is why it's important to numerically, before getting into this kind of anomalous diffusion models, I want to introduce the regularizing a trajectory. This is a very straightforward algorithm. Assume that I have a trajectory that, think about it like this. If you are measuring the position of a particle with a frequency, but your camera is dropping frames, you don't have the data for every time, okay? You have kind of irregular sampling, which are the blue dots. So here you have data for particle position, or you can model, think this, for example, as an example, I can give you this kind of bioluminescent particles in the ocean. They kind of shine light once in a while. They're fluorescent bacteria, right? But they don't do it regularly. So you kind of know where they are when they blink light, right? So you don't really get a linear data in terms of time resolution. So you know that the particle was here this time, particle was here this time, and then for this much amount of time, 20 seconds, you have no idea where the particle is. So it's gap. So these kind of things. Of course, this is very difficult to characterize, because all the things that they are making with these arrays, we are assuming that the time difference between each element, consecutive element, is the same, right? If you have a trajectory that the time difference between each data point is not the same, it's very difficult to analyze, especially if we have two different classes of trajectories that have different kind of time resolutions. It's very difficult to compare statistically. That's why we need to have a function numerically to go from this blue circles to orange crosses, so that we kind of regularize. What we do here is that we kind of go linearly in time and sample whatever the point is. I mean, we make a linear kind of approximation for each point in time that we calculate the position kind of. Is this clear? So we kind of regularize the trajectory in time. We kind of have the same trajectory, but we can now have a much easier calculation in terms of square displacements or in terms of other things in the trajectory. So this is kind of just a numerical method, and there is the description how to make it in steps in the lecture notes. And of course, now we can maybe talk about different kind of models for modeling the anomalous diffusion. So before we get into that, the anomalous diffusion is described in two different categories. So the main thing is that if I have a regular diffusion, my mean square displacement is scaling linearly with time. Okay, this behavior is represented here with the black dashed line. If it is scaling larger than linearly, which means that it's going, this slope in this plot is higher than 1, which means that the particle is super diffusive, which means it's going displacing faster. Its displacement is scaling faster than t with time, square displacement, which means that it is a super diffusive particle. Example, all the ballistic motion, all the things that are moving with constant velocity, people, animals, these can be examples of super diffusive. They kind of make diffusive motion as well, but most of the time they kind of decide and go in a certain direction for a while. And when we make that, this is kind of a ballistic motion, so that our square displacement is scaling with t squared instead of t. So this results in a super diffusive behavior. And anything that is falling below this trend, where the slope is less than 1, is called subdiffusive. And this means that anything that is not able to perform free diffusion due to some obstacle, limitation in movement, anything like that, any kind of this system is showing a subdiffusive behavior. Is this clear, the difference between superdiffusive and subdiffusivity? For example, a motion of an optically trapped particle is what? Subdiffusive, right? Because it's not able to perform. It's in the short time scales, it's diffusive because it has not felt the optical trap yet. It's displaying a little bit around. As soon as it goes a little bit far away from the equilibrium, there's a strong force calling the particle back to center. And then it cannot, the mean square displacement cannot scale linearly with time. It is kind of saturating. All the motions of organelles, for example, inside the cell are subdiffusive. Although they are making the diffusive motion inside the cell, they are kind of limited by the cell membrane. They cannot go out of it. All the motion of bacteria that are swimming, what is their motion? Is this subdiffusive or superdiffusive? Superdiffusive, right? If a particle that is passive, that is just diffusing around with collisions of other molecules, that is a regular diffusion. But bacteria is doing more than that. It is swimming, making an effort to go into a directed motion that creates a superdiffusive behavior. Okay, and this results in, actually, if you check the motion of the bacteria, that you will see micro swimmers section in chapter 9, I think. And you will see that their motion is also superdiffusive. Okay, now we kind of know what superdiffusive and subdiffusivity is. So now we are going to give an example of an anomalous diffusion model, which is one of the most common ones, is the scaled Brownian motion. How do we obtain a scaled Brownian motion trajectory? Is the following. So it's very straightforward. You simulate a Brownian particle, just like we did in the previous examples, like a diffusive particle that is going around randomly in one dimension or two or three, doesn't matter for this case, it will give you the same kind of, it's not different between different dimensions. And let's see. So you get kind of a trajectory of the particle, right, as an array. And you have the time also. And this is like, let's say 0, 1, 2, 3 times delta t, whatever my delta t is in the simulation, right? So I have these are the times, that is corresponding times for my trajectory data points. And if I take the mean square displacement of a linear diffusion simulation, what will I get in terms of the exponent? How does it scale with time? T, right? This will scale with the mean square displacement of this x will scale by t to power 1, right? But what I can, how I can change this very trivially is that I can simulate this. After the simulation, I can rescale my time. After I make the simulation, I rescale my time to another time, Ts, that is t to my power 1 divided by alpha. And immediately what will happen is that this will start scaling with t to power alpha, because I took the whole time array and scaled it with to the power of 1 divided by alpha. You understand the mathematical point? And then what we end up is having kind of these kind of trajectories. You see that the blue one, when alpha is equal to 1, is for the scaled Brownian motion, is the regular diffusion. It's very standard. It's looking like a trajectory from a random walk or from any other distribution steps that I was showing in the previous lecture. And if alpha is smaller than 1, which means that the time scale, the displacements are going to, what will happen is that if I'm taking the steps, let's say, let's start easy. If alpha is equal to 0.1, sorry, 0.5, what will be 1 divided by alpha? 2, right? And I'm going to scale my time array by taking the square of it, 0, 1, 4, 9. It will look like I'm taking the same steps in a longer time, right? It will look like over time I'm moving less and less, right? That's what you observe here with the green. The steps look large in the beginning compared to the steps at the end. Of course, this is statistics. I mean, if you plot a longer trajectory, you will see better how it's going like. And of course, the alpha 1 case, the steps are always the same, the distribution of the steps. But if alpha is equal to 2, which means that I am scaling my time array, which taking the power of 1/2, which means that I will rescale my time like this. So my time array will be 0, 1. Of course, I am regularizing this. These are regularized trajectories. Okay, and you see that I am taking the similar steps between these, but less time has elapsed between steps over time, right? If alpha is larger than 1. This means that my steps are going to get larger and larger over time. Is this clear? And of course, after I do this, I have an array of time that is not regularly sampled. I just need to apply the regularization that I was showing you in the previous slide. And then you end up with these trajectories. And this is how they look like in 2D, roughly. And what we can now do is that we can try to see if they are ergodic, which is that I try to see what is the ensemble average and time average of the mean square displacement of this motion. Of course, here the characteristics of time motion is changing with time significantly, right? It's not the same if I'm taking the motion in the beginning of the array and taking an ensemble average, or if I'm taking a very long single trajectory and making a very long time average. It becomes significantly different from one another. And that's what we see here. This is for alpha is equal to 1.5. Of course, if alpha were to be equal to 1, this would be the regular diffusion, and we would end up again the same identical results. I am here showing you that if alpha is not equal to 1, the scaled Brownian motion is if for alpha 1.5, it is super diffusive. This alpha is, by the way, the same alpha that you get at the. So this is alpha 1.5. Then which one is giving us the right result? Which mean square displacement measurement is giving us the right result? Looking at this plot. Ensemble average, right? It's the one that is scaling correctly according to the theoretical prediction. Is this clear? This is how we kind of model the motion of super diffusive or subdiffusive particles. So importantly, scaled Brownian motion model can model both super diffusive and subdiffusive, depending on your choice of alpha, if it's larger than 1 or smaller than 1. And the next one I will tell you about. There are more models, by the way, than the ones that I'm introducing here. But you can go and investigate. I will not cover all of them because it will take just way too much time. Here is another one is continuous time random walk. And here what we do is that we operate a certain amount of displacement and we wait before we walk again to another random amount. And the waiting times that I have is power law distributed. So it can be really something very short or something super long. And this represents actually very well the motion of all these proteins, enzymes, or organelles in the cell, because most of the time they are stuck and they are not allowed to move. When they are required to function, then they can displace, they can operate, they can go to another place, they can become free, all this kind of stuff. This is a very powerful model that is used also to simulate systems inside a cell, for example. And here what we do is that we kind of make a random, again, Brownian motion trajectory. And instead of making a linear time array, we this time have a waiting time array. What do we wait between each trajectory points? Okay. And then the waiting times are calculated by this formula. And here again, alpha matters. And here, importantly, for the continuous time random walk, alpha cannot be larger than 1. This is a model that is describing the subdiffusive motion only. Okay. And here you can see again that the differences, if you are only taking the average over different realizations or average over a single long trajectory, are different from each other. And this is an example how they look like in 2 dimensions. Is this clear? Yes. Because we are making the regular Brownian motion, right? And then in between them, we are waiting. We are kind of stuck for a while. And these waiting times are power law distributed. If we were waiting the same amount of time, that would actually not be, because what does it mean if we were waiting the same amount of time? It means that this alpha would be what? Would be 1, right? Which means that my waiting times would be uniformly distributed. So this creates a kind of regular diffusive behavior. It can go all the way up to regular diffusive, but it can never go to super diffusive. Because if your waiting times, if alpha is very small, your waiting times can be extremely long. So you are stuck at one place for a long time, which makes you subdiffusive. You cannot diffuse. But of course, if your waiting times are shorter and shorter, importantly, if they all become the same, then it's kind of a, again, regular Brownian motion. And another model is called Levy walk. This is a very interesting kind of anomalous diffusion model also, because it's modeling most of the motion of animals that are foraging food. So they're looking for food to eat in nature. It can be fish, it can be birds, it can be insects, anything that you can name. And it's modeled like this. So we we assume a constant velocity. This can be assumed as a swimming velocity, flying velocity, walking velocity, whatever. And then we kind of generate walking times with this kind of distribution. This is a, I mean, don't get confused by this weird formula. This is because mathematicians want to make sure that when you take this formula, the exponent of the mean square displacement of the resulting diffusion trajectory is going to scale with alpha. That's why they come up with kind of this kind of formula. So that is to make sure that this alpha, when I choose my alpha, I want my mean square displacement to scale with t to power 1.5, and then I can put 1.5 here, and it's analytically made in a way that convenient that if I use this formula, I will get kind of that kind of scaling. Okay, it's nothing else. And all of the formulas you see here are I is a uniform random number, which means that it's scaling from its rand, mathematically speaking, so uniformly distributed between 0 and 1. And you kind of run in one direction you choose for a while with, so you calculate your next time like this. And here WI is not a random number, but it's a direction. So if I am in 1D, it can be plus or minus 1. If I am in 2D, it can be a certain direction, which way I am going, okay? You see here, 2D trajectories and 1D trajectories are significantly different from one another. You cannot just make a 2D trajectory of a Levy walk and take the component, one component, X component of this trajectory, and assume that it's a 1D Levy trajectory. It's not. Because here you can only have one velocity, up or down, the same. But here you can have different velocities because you may be going in different directions. So the motion is modeled differently in 1 dimension and 2 dimensions. It's kind of important. And then you kind of calculate. So these are kind of times that your walking times. And here are 2 different examples for alpha is equal 1 and alpha is equal 2. Is this clear? So these are all written in the lecture notes, so you don't need to write them down. And also, by the way, we are uploading this in all the slides in the files in the course. So whenever you need it, and right under the Zoom link on the on the Canvas homepage, there is a YouTube playlist. So there is the recorded lectures, all of them. So you can go and watch them again. So you don't need to really stress taking notes. Okay, just try to understand it. And then all the times are the time scales, total time scales are the cumulative sums of these walking times, right? And then I might kind of make the regularization of the trajectory. And then this is how it looks like in 2D. And you see that this is a lot more ergodic, although it's still weakly non-ergodic as mathematically defined. But the trajectories between different ensemble and time averages look very similar to each other. Of course, there are different kinds of anomalous diffusions for different systems, but a very standard example of an anomalous diffusion is speckle fields. If you have a speckle light field and a microscopic particle, since higher intensity light regions attract particles, just like the case of optical tweezers, this particle may get stuck in one single area. You see that the trajectory of this particle, green, is stuck. This is a, so these are experimental data. So that you see that here is a higher intensity area, the particles get trapped there because of the laser power is trapping them there. And if the laser power is smaller, what will happen is that particle will get trapped here, but sometimes because of the probability, they might be able to escape from here to another one, here to another one. So this is kind of a subdiffusive case. And if we have no power, the particle will just diffuse around randomly. And this will be a regular diffusion. And this can actually be modeled according to the average force acting on the particle with respect to laser power. And the standard case, when the power is very small, you see that it's when the laser power, average force acting on the particle from the laser, is very small, it's almost freely diffusing. And as you increase the laser power, the particle gets more and more trapped. This creates a subdiffusive behavior. This is also anomalous diffusion. But these kind of environments can be used very wisely in order to sort microparticles, for example. Because how does it affect this light field, the particle? What particle parameters does it depend on this effect? This trapping effect depends on particles material, particles refractive index, particles radius, right? So you can use these kind of trapping patterns to sort different microscopic particles of different kinds or different radii. Here, for example, the first exercise, they kind of apply a field. This is numerical results, and they kind of apply a field downwards, but the green particles are kind of this like a thought experiment, let's say, and this is a microfluidic channel, and they can kind of separate two different kinds of particles. And also here, another similar idea is that you kind of have a speckle field that is tuned in a way that it can trap this kind of black type of particles that have different refractive index than the green ones. And it is kind of designed in a way that once in a while, green ones are also getting trapped. But if you have a long enough speckle field, they will totally differentiate from one another. This kind of environments can be very smartly used to distinguish different kind of the particles with different properties. Is this clear? And also, one last example is that, of course, it is most of the time we create models and try to understand the different kind of anomalous diffusions with respect to these models, right? But sometimes we have an experimental trajectory from the nature. We have a trajectory and we try to understand what is the best underlying model that is characterizing this trajectory, right? And of course, we can have this kind of trajectory and we can kind of get a trajectory and we want to understand which underlying anomalous diffusion model is most strongly representing this trajectory that I have. And this is very important challenge in the field of anomalous trajectory. Just last year, there was a challenge that was named Anomalous Diffusion Challenge that is called the Andy Challenge. You can have a look if you are more interested. And this actually has been, the challenge has been, it's essentially a data analysis contest. So they have trajectories and the tasks are find the anomalous diffusion exponent of a trajectory, this alpha I am talking about, if it's subdiffusive or superdiffusive. Find the underlying anomalous diffusion model, the scaled Brownian motion, Levy walk, and continuous time random walk I told you about in this class. There are two other models, and make a segmentation. If there is a trajectory and the particle is changing behavior at a particular spot, where does this change occur? And what were the models before and after the change, kind of. These kind of things were also studied, and there is actually a workshop coming in a few weeks. Actually, we have to go, unfortunately. I say unfortunately because it's colliding one of the homework correction sessions. Is there any questions about this lecture? Okay, so every group has to, just a reminder, every group has to arrange a meeting, tutor meeting with your tutor about your project. And you're going to send, or most of you have already sent, the project proposal. If you haven't done it, do it immediately. This is for us to have an idea, read it, and have an idea about what you want to do, so we can give you useful advice. Okay, we are going to have a meeting with each group for about 45 minutes. So if you give us more materials of what you are thinking about, we can give you more valuable feedback. Okay. Thank you very much, and the lecture is over.
How about if your proposed project is like